Welcome to another Biopsych lecture. In this, I'm going to spend a little bit of time talking about these guys. I'm going to talk about some of the techniques we use to study these and some of the basic neuroanatomy we've learned from that. Neuroanatomy is the scientific study of the nervous system's structure. That's it. How do we study neuroanatomy? Well, we break things up. We can study brain structures themselves. We can study individual neurons. We can study clusters of neurons in the central nervous system. Those are called nuclei. Don't confuse it with a nucleus of a cell, though. It's different than that. Together, these make up the gray matter. We can also look at connections between structures. This is very important to understand what connects to what functionally. These are so-called fiber tracts. They're just bundles of axons. We'll talk a lot about these moving ahead. As you know, the axon is coated by myelin, which is fat. If you look at a cross-section of a brain, these appear white. So gray matter equals cell bodies, or soma. White matter equals fat myelin, which equals axons. Now, we study all of these separately. There's different techniques to study sort of the larger structures. You can kind of use your naked eye, but mostly we'll need a microscope in order to see things like individual neurons. Fiber tracts are a bit harder to study, and figuring out where they go is a lot more involved. We know less about that in a human than we do these other things. I'm going to start out with, at least from a neuroanatomical perspective, what's probably the more useful techniques. For these, we require a non-living brain. So you have to kill the subject or wait for the subject to die in the case of a human. Imaging the non-living brain. These are great techniques that have been used since early neuroscience. One thing we can do is just gross dissection. Gross means what you can see with your eye, not gross in the other sense. This is a mid-sagittal cut of a human brain. Okay, there's some stuff I'm going to expect you to know. We have the cerebral cortex. That's all this stuff. You can see here we have peaks. Those are gyri. We have the valleys in between. Those are pronounced sulci. Some people say sulci. We'll go with sulci. Here we have the cerebellum. You can see white matter here versus all around it, which is grayish, a little grayish. Okay, so the white matter would be bundles of axons. The gray matter would be cell bodies. Here we have the corpus callosum. In the mid-sagittal section, that's usually the most prominent feature. Here we'll have the thalamus. Here we'll have the hypothalamus. Hypo means below. Right in here. Here we'll have a structure called the pons. Here we'll have the medulla oblongata. I'll often just call it the medulla, but the correct term is officially medulla oblongata. You have a lot of medullas in your brain, in your body, and a few in your brain. But this refers to only one structure. So there's only one medulla oblongata. Okay, but we can see some things, but can you tell me what this part of the brain does? No. Can you tell me what this part of the brain does? No. These tell me nothing about function, right? They just tell me about structure. Okay, so it's fairly limited in how useful gross dissection is. At the microscopic level, though, we can learn a lot more. So this is a microscopic level. This is a central nervous system stain with something called Golgi stain. Golgi stain, I mentioned before, is an awesome stain. Completely fills about 1% of neurons. We don't know which neurons, though. It seems to be random. We don't know whether certain populations take it up more than others. But if you look at this photomicrograph, here we can see the soma, and we can see processes coming off. Now, if I look, I've got a whole bunch of processes. This is clearly a multipolar neuron. Which one's the axon? Well, based on this, you can't really tell. Okay, so one thing about Golgi, its limitations, you can't see what's inside the cell. So I can't tell which of these is the axon based on this photomicrograph. I know we had to process. I know there's one axon, all the rest are dendrites, but I don't know which is which. Okay, so I'm going to tell you a little bit how we get to this stage. You don't just pop a brain out and get this. We have to do some stuff to it. In order to do microscopic analysis, the first thing we have to do is preserve the tissue. If you leave anything out for a long time, it basically just rots. Brain tissue is no different. In fact, the brain is mostly fat. I shouldn't say that. Brain has a lot of fat. So that when it breaks down, it smells horrible. It is one of the worst smells I've ever smelled. So the first thing we need to do is do a perfusion, which is basically drive preservative through the circulatory system. So I'll take a needle and I'll put it, say, in the heart. And then I'll pump formalin or glutaraldehyde or another preservative through the needle into the heart and then through the animal circulatory system. You saw the pictures for the blood-brain barrier. There's tons and tons of blood vessels. So when this gets to the brain, it's a great way to deliver the preservative to the brain, and it helps ensure that the brain is in good shape. The one preservative I want you to remember is formalin. Formaldehyde, either one. They are slightly different, but basic same structure. What these things do is they kill bacteria and cross-links proteins. So it's going to get rid of the bacteria that would normally break down the brain, and it's going to take any proteins that are there and sort of stick them together. That's going to keep things from moving around. So it's going to maintain the structure. Now, once I'll have this, I'll have a block of tissue. Okay, so I have a hunk of brain tissue. It won't rot anymore, but I can't see anything. If I want to see anything, I'm going to have to cut it into thin slice. Very thin slices. The reason I have to do that is so that when I put it under a microscope, the light can pass through it. It's got to be that small. So usually what we do is we'll freeze the tissue. That makes it nice and firm, and it makes it so the microtome can cut. So a microtome is an instrument that cuts the brain into thin section. Okay, so we're going to get very thin sections, so thin that light passes through them easily. Don't worry about these other two for right now. Okay, in order to do that, like if you've ever taken a tomato or something that's really super ripe and you've tried to cut it, it's really hard to get nice, even, thin slices. But if you have a tomato that's not quite ripe, it's actually easy. So the firmer the thing you're trying to cut is, the thinner the slice you can do. So freezing it is going to make it very firm so I can get a very thin slice. Typically, we're talking something like 20 to 40 microns, 10 to 40 micrometers for an average slice. This is just showing you how we cut it. This is a microtome. Here's a very sharp blade. This sample is going to go here. I then move this forward. As I move it forward, this sharp blade is going to cut through my sample, and I'll be left with a little hunk of tissue on here. I take a paintbrush, I pull that off, and I put it into saline. There it'll expand out, and then I can mount it on a slide. Okay, it's pretty basic. Now, once I do that, I have a very thin slice. It's translucent, meaning you can see light through it. Okay, so I have a thin translucent slice. Shows me nothing. If I want to see something, I have to stain the tissue. There are hundreds, if not thousands, of different stains out there. And which stain is best depends on your questioning what you want to do. If I'm interested in studying connectivity in the cortex, for example, here we've got human cortex, 180 microns. So these are very thick sections, much thicker than usual, cut on a cryostat. All right, and this is there's a kit you can buy if you want to do Golgi stain. Okay, and this is the kind of material you would get. So remember, this is Golgi stain. So you know you're only seeing about 1% of neurons here. But what I can see is the cell body plus all the processes. And again, if I'm really good and I can look on higher power, I can trace these for a long way. I can figure out where they're going, where they're coming from. Okay, so again, you can look here. This reason you can tell that this one is an axon is because you can see it connect to another cell. So that tells you this is the axon. Everything else in this will be dendrites. Okay, so on high power, if you're lucky, you can follow this and see where we actually make a synapse. Okay, so Golgi stain is going to be great for letting me look at shape of neurons and maybe some connections. Is this going to be good for counting the number of neurons? If I want to know how many neurons are here, can I tell from Golgi? No, that's a limit of Golgi. It's not good for counting the number of neurons. All right, here, what kind of stain is this? Hopefully you recognize that right away as Golgi. I expect you to be able to recognize that. Over here we have a different stain. This is called H&E, hematoxylin and eosin stain. Don't worry about that. But if you look, these two pictures, even though they're taken from the same type of tissue, look really different. Okay, because the stain has different goals. It's going to show us different things. So H&E is sort of your standard pathological stain that they'll use when they're doing medical diagnostics. Right? Here we can see a cell nucleus, you can see a cell body, right? But the shape of neurons, what we would see, is not real well emphasized in it. So consequently, like neuroscientists don't use H&E stain. We use Golgi and something called Cressyl Violet. Okay, so I want you to remember Golgi and I want you to remember Cressyl Violet. Cressyl Violet is going to stain Nissl substance, which is concentrated in the nucleus. And so here, if you look at this, this is going to be a coronal section of a mouse brain. Sorry, this is a rat brain, my bad. Of a rat brain. And if you look, you can see some places where dark, very, very dense. So here, for example, this is a very high concentration of stain. That means a lot of cells. Okay, so you can see lots and lots of cells here. Look over here though. In this area, it's pretty light. That means there's only a few cells there. We can look, this is actually going to be part of the hippocampus. Okay, so we can see there's clusters of cells, very dense, surrounded by less dense areas. If we look here, you can see this round spot, right? So here I can trace the outline of this cluster of cells. That's a nucleus. This is going to actually be the ventromedial nucleus of the hypothalamus. All right, the important point to note, though, is that you can see this cluster of cells compared to the area around it. Okay, so crystal violet stains every cell. So this is going to be good for cell counts. Okay, again, probably the two most common stains you see in neuroscience. Crystal violet is exceptionally common, by far used the most. But Golgi is also used quite a bit. Here's just some more examples. This is crystal violet staining of hippocampus. We've got some dentate gyrus here, which is a subregion of it. If we look on the high power, you can see each dot represents one cell. Okay, so when we look at all of these, you can see the cells. Okay, so crystal violet is great for letting me see individual cells. This is a coronal section from a rat. Here, if you look, I've got the anterior commissure, third ventricle, optic chiasm. Those will mean more later after this chapter. In panel A, I've got a male. In panel B, I've got a female. And if you look, right, you can see this so-called SDNPOA being pointed at here. So in the male, we can draw something like this. Okay, that's the SDNPOA. In the female, we can draw, it's a little harder, right? But we can draw something like that. If we look using crystal violet, that volume of the nucleus, males are greater than females. Under high power, we can also count the number of neurons. Male also greater than female. If we look at the size of the neurons, male greater than female. So there's a lot we can tell. Now this is telling us about the structure. It's not telling us anything about the function yet. But we can get a handle that maybe this SDNPOA, by the way, SDNPOA, sexually dimorphic nucleus of the, I spelled nucleus wrong, sorry. Nucleus of the preoptic area. You can see why we call it SDN. And really, we should also add of the hypothalamus. Okay, but we can see that it's larger in males than females. And if we're going to look for function, it might be a place where we want to look at behaviors where males and females differ, because the volume of the nucleus is different between the two. Okay, that's something we can do with crystal violet staining here. I'm going to talk about a very important technique called immunocytochemistry. This is going to use antibodies against specific proteins. Okay, so things like enzymes, like TH, tyrosine hydroxylase, receptors. We'll say nicotinic acetylcholine receptor. These are all proteins. So if I take some human nicotinic acetylcholine receptor and I inject it into a mouse or a rat, that animal is going to make an antibody against it. Okay, so I'm going to take my protein of interest, let's say in this case TH. I'm going to take TH and I inject into an animal. Animal will raise antibodies against it. It thinks it's foreign. So now it makes antibodies. I can then isolate antibodies. Okay. So now I have antibodies from my animal against tyrosine hydroxylase. So if I take a tissue, let's say I got a brain slice, and let's say I have some TH in these areas here. If I take my antibody and I soak my antibody with the brain, what's going to happen? Well, these are going to float around, and wherever my protein of interest is, they're going to stick. Okay, so that's what this is showing. My primary antibody, in this case, let's say it's against TH, anti-TH. It's going to float around in the liquid. Wherever I have TH, though, it's going to stick. Okay? Now, I can't see an antibody by itself, so I have to do a little biochemical mumbo-jumbo. Somehow I have to label this. In some antibodies, I can actually take a fluorescent marker. So a fluorescent molecule, and attach it to this antibody. Okay, so these will glow when I give them fluorescent light. Okay, so I take my tissue, I let these antibodies float around. Some of them are going to bind to the TH and stick there. I then rinse everything else away. So all I'm left with is antibodies stuck to my protein of interest. I shine a fluorescent light on. Wherever I see glowing, what do I have? An antibody. If an antibody is there, why is it there? Because it recognizes TH. It recognizes my protein of interest. Okay? Now, this isn't particularly sensitive. There's an improved way, which is what's shown in this cartoon. Okay, it's the so-called ABC method. What I can do then is take, after I do my rinse, I've got my antibody stuck. My primary antibody, this one here, the first one, isn't labeled at all in this case. What'll end up happening is I've got another antibody against the first antibody. Okay, so this is a second antibody. This is going to recognize the first antibody and stick to it. Okay, and these black dots represent markers. Okay, things that I'm going to be able to see. I then incubate with another chemical and run some biochemical tests on it. This is a big enzyme that catalyzes a reaction that ends up with a product I can see. So we take the second antibody, it binds to this. We then take this ABC, this avidin biotin enzyme complex that sticks to that second antibody. I then add substrate, and there's a chemical reaction that occurs. At that chemical reaction occurs, I get a brown spot. So wherever there's a brown spot, there's this enzyme complex. Wherever there's this enzyme complex, it's there because of this second antibody. Why is the second antibody there? Because of the first antibody. All right, bottom line, this lets me amplify the signal a lot. So I'm using an antibody specific to a protein. I'm doing some biochemical mumbo jumbo so I can see where it is. and it'll turn brown in the cells. Okay. I can also kind of mix and match things too. This is a simpler one. So here I've got my antibody against my protein of interest. Here the second antibody has a glowing. So this is a fluorescent. Okay. The point of this first antibody, the so-called primary antibody, is against my protein of interest. This second antibody, everything after is just amplification, amplifying the signal. Here it just shows one bound to this, but actually we can get 10 or 20 of these molecules bound to it. So we'll end up getting brighter and brighter. All right, so what does this end up looking like? Remember I showed you that Avidin ABC method where it's an enzyme and we get a brown product? Here's what we see. So here is a cell. This is a GnRH cell with the primary antibody versus GnRH, which is a small protein. Okay, the primary antibody bound there. Then those other things bound to it. I ran my chemical reaction and I can see where it is because it's a brown product. We can also look for other substances too, and you get all kinds of beautiful pictures. Okay, now I'm going to do this one last time. It can be a little confusing, so that's why I'm doing it again. I can have my cell with different proteins on it. I have an antibody against protein A. So here we're looking at protein A. Okay. This antibody will stick to it. And we've got a fluorescent staining tag. So here I can see where this antibody is. Now, because there's only one of them bound to it, it's not real sensitive. I don't see a whole lot. I can amplify my signal. Let's say the antibody was made in rabbit. Okay? So I took my protein A, the protein I'm interested in, I injected it into a rabbit. The rabbit made antibodies against it. Okay, so I take my tissue, slice it thin, I soak it with a solution with all this rabbit anti-A. Here, only one molecule is shown. In reality, a few molecules would bind to it. Now I can use a similar principle where I can inject a goat, say, with rabbit immunoglobulins, with these antibodies. So now the goat will make antibodies against the rabbit stuff. And I can add a fluorescent marker to that biochemically. Here only one is shown, but again, in reality, 10, 20, many goat anti-rabbits join and bind to the rabbit antibody. Okay, so even though only one's shown, it'd be many. So instead of just one being fluorescent, now many hundreds would be fluorescent. And so we'd see a brighter signal. Okay, so you can get all kinds of pretty slides. This red is neurons, the white is axons. Again, so we can label for more than one thing at once. We can label many things at once, as long as we have different antibodies. Okay. I'm going to end on that note for a moment.